Right, so we are officially moving into protein expression, and we're going to start with chapter 13, which is transcription in the Pierce text. So, transcription is the first part of the protein expression process. The second part is translation, which we'll catch in chapter 15. So the way that we are going to work through this chapter is, first of all, we're going to talk a little bit about what protein synthesis is and the role of RNA and transcription. And we're going to mostly focus on the RNA structure being the primary source of genetic information early in life. Then we're going to move into talking about how the mechanism of transcription, so how the DNA template is required for transcription as it progresses through initiation, elongation, and termination. And then finally, we're going to talk about differences that we see between prokaryotic and eukaryotic genomes in regards to transcription. All right, so again, protein synthesis or protein expression is really a two-step process. The first part is going to be transcription, and the second is translation. So, transcription is actually creating a new message based on the DNA template, and then translation is taking that new message and translating it into protein code. So, this is how we really get the transfer of genetic material from the genotype in the DNA form to the phenotypic expression. Okay, so just to set this up a little bit, what we've got here is our DNA in a double helix strand, and the DNA is then going to be activated, or a portion of DNA is going to be activated, and that's what we really start to call a gene or a loci. So, the first part of that activation process is transcription, and the first step is the gene is actually going to be turned on. And so, very similarly to DNA replication, the DNA is going to unwind at the start site. RNA polymerase is going to come in, sit down on the promoter region of that gene, and transcribe it, or copy the gene. And then that copy is going to be called the messenger RNA, abbreviated as mRNA. Translation is going to be where mRNA is going to be translated or read by the ribosome and made into a protein. So think of this as just like when we translate languages. So we're taking one language, translating it into another language that can be actually expressed. So here we've got the ribosome in the translation process that actually does all of the work, and it will actually bring in transfer RNAs and start building those amino acids together into a polypeptide chain. So RNA was really the primary um, the primary material that of life is what we think. So this is evolutionary genetics has really shown us that RNA was a big player as we started the evolutionary process. So we know that life requires two main functions. The first is to store and transmit genetic information for the reproductive process. So that's going to be meiosis and mitosis. And the second of the two function is that life really requires a chemical transformation of some sort of catalyst that drives that process or that protein expression. So, a lot of our early evolutionary biology in life centered on RNA as being the catalyst of or the genetic material. And eventually, as we evolved and or the environment progressed around us, DNA really replaced RNA as the primary carrier of genetic information. And that was because DNA honestly was more stable. And we talked about why it was more stable in chapter 10. So I encourage you to review that if you don't have it on the top of your brain anyway. And then DNA was faithfully replicated through the process of DNA replication, which you now know all about. So RNA's structure has three major differences compared to its DNA counterpart. So first of all, in RNA, uracil, or U, is going to replace thiamine. And the second major difference is that the hydroxyl group is going to be on the 2' carbon of the sugar rather than the 3' carbon. So that 2' carbon you can see here, and we looked at that difference between ribose and deoxyribose. So the sugar's changing as well as that uracil base. And finally, RNA is going to be single-stranded, not double-stranded like DNA. So three main differences between RNA and DNA. Uracil replaces thiamine in RNA. The hydroxyl group on the 2' sugar is present, and RNA is going to be single-stranded. So one of the minor differences of RNA that is really unique to how the process of transcription works is the secondary structures associated with RNA. So we talked in chapter 12 about secondary structures being really bad. If DNA became separated from its complement, then it became very, very unstable. And even during the DNA binding process, those single-stranded binding proteins actually help to stabilize that single strand of DNA. But with RNA, we count on secondary structures to form, and this is going to help with the signaling process. So keep in mind, primary structure really has to do with the sequence of nucleotides, and secondary structure has to do with how those sequences orient themselves to bind up with one another. And that's going to become more clear as we talk about termination, both in transcription and translation. So this table in your book is really a good way to look at the differences of DNA compared to RNA. So both structures are going to be composed of nucleotides, but the sugars change. So deoxyribose is going to be present in DNA, whereas ribose is going to be present in RNA. And then the bases are going to be the same, except T is going to replace, or U is going to replace T with RNA. So there's several classes of RNA, or ribosomal nucleic acids. So the first one that we're going to talk about is ribosomal RNA, abbreviated as rRNA, and this makes up the ribosome. There's a little bit of protein associated with it, but primarily it's going to be made up of RNA material. The second major class of RNA, and the one that we're going to talk about a lot because it's actually the product of transcription, is messenger RNA. So messenger RNA very much carries the message from DNA to protein. It's the middle step. A third type of RNA is a pre-mRNA, or a pre-messenger RNA. And we believe that these pre-mRNAs were really the early classifications of RNA before we evolved into using DNA as the genetic information. So in eukaryotes, we really see pre-messenger RNA present. We don't see it present in bacterial genomes, and pre-messenger RNA is going to require a processing from to mRNA. So it's going to be the structure that's actually formed first, and then it will be edited to get the actual true messenger RNA. And finally, the fourth one that we're going to spend a lot of time talking about in chapter 15 is transfer RNA, or tRNAs. And this transfer RNA is going to take one amino acid and bring it into the ribosome as it starts to pair up a polypeptide chain. So again, just know about transfer RNA, but we're going to discuss its function more in chapter 15. So your table 13.2 in your book really gives a good synopsis of the similarities and differences between all the RNAs. And there's a couple on here that I didn't mention in the previous slide, like SNO RNAs, silence RNAs, or Peewee interacting RNAs. And one of the new ones that is really starting to take a front seat as far as popularity in genetic research is CRISPR RNA. So I encourage you to watch this video about CRISPR RNA from the Broad Institute, and they will explain to you what CRISPR RNA is and why it is so important as we're talking about genetic diseases and curing some diseases within human and animal species. All right, so these different classes of RNAs are going to have a lot of different functions, but again, this slide really depicts out the RNAs that are going to be involved in transcription and the ones that are going to be involved in translation. So again, messenger RNAs, ribosomal RNAs, and transfer RNAs are probably the ones that you learned about in biology, and these are going to be transcribed in both prokaryotic and eukaryotic cells. The pre-messenger RNAs, small nuclear RNAs, microRNAs, and small interfering RNAs, as well as a few others, are going to be produced only in eukaryotic cells. So, just as a fun side note, or I think it's fun, the silence RNAs, or the small interfering RNAs, is we actually worked with in the lab, in my graduate student lab, when we were working on a breast cancer cell line. So the whole point was to test these different sequences of silence or small interfering RNAs to see if it could shut down the tumor characteristics that have to do with the breast cancer cell line. So those are sum up the basic classes of RNAs. So just keep in mind the ones we're going to concentrate the most on are messenger, ribosomal, and transfer RNAs, and those are going to be in both our prokaryotic and our eukaryotic.